then you can trim the adapters and low quality basis using the tools such as Trimomatic or Kuta-Dapt. Then remove contaminant sequences also. And these are few minutes work only. So there is no need to be afraid. And what I'm telling the steps, they are manual steps. So the next part is read alignment. Before you proceed for read alignment, you need to choose the complete human reference genome or the organism which you are working on. For that particular organism, you need to choose the complete reference genome. And this is very, very crucial. If you are choosing the file of reference genome, which only includes the chromosomes, all the chromosomes, and they do not include, for example, the mitochondrial genome sequences, then if there is no mitochondrial genome sequence present here, you will never be able to map any gene which are present in mitochondria and not present in any other places. So it is always clever idea to select the human reference genome which includes each and every sequence of all the genomes which are present in human. And then once you choose the reference genome, then use this command if you are doing manual like NGS RNA-Seq analysis, then generate the index file for the reference genome. And this will generate the 6 files. There will be 6 files generated from the one human reference genome, and the command you need to use this command POTI2 build. And this large index, so this you can select whether you want this option or some other option, it's up to you. So there are several options here where you are writing large. And then minus index minus F. Minus F is input file, telling that input file is named as hg38.fa. Then this hg will be the initials for all the 6 output files. So this is for output file nomenclature. Now, second step is to align the reads to the human reference genome. And this alignment will require these 6 files of the human reference genome which you have indexed. And the way of using the command, if you are using POTI2, which is the most frequently used software for alignment with the reference genome, and then you need to use the command POTI2 minus P, that means how many processors of your computer you want to use. Larger the number of processor, faster the alignment process. And then I have selected here local alignment. That is why this is the option written here, local. Then you need to instruct that this is minus X. That means where is the file and what is the file name. The file will be, let's say, for example, B1 means these H. So here you need to write HG. If you are generating the output file of reference genome with HG, then write this HG. Then minus 1 means R1 file, minus 2 means R2 file. After writing minus 1, you need to write R1 file name, and then after minus 2, write R2 file name. Then minus S is SAM file. That means the file alignment file will be stored in this SAM file. and then you need to assess the quality of alignment. This step takes in general more time than all other steps. So this is where you need to have powerful computer, or you need to wait for the output for longer time. All other steps are quite fast and easy. Then once you have done these things, then there are two more steps where you need to convert the SAM file into BAM file, and then BAM file will be converted to sorted BAM file. And these BAM files are just suppressed and properly indexed file of the SAM file. So SAM file is always much larger than BAM file. So BAM file is easier to store and process for the next steps. So after converting the SAM file to BAM file and sorted BAM file, you have now like transcriptome, assembly, such things you have to go. And these are something which I will say are done in the previous file, where you are generating the SAM, converting the SAM file to BAM file to sorted BAM file. So this is just extra thing. And for this purpose, some people are using a string tie or cufflinks, but you don't need to use anything. Just go for these bow tie things, which will be much helpful for you to proceed. And in converting BAM file also, you need to use, instead of like, bow tie, you need to use something called SAM tools. And SAM tool is used for multi-purpose, but one of the most common use for SAM tool is to convert the SAM file to BAM file. So this is also quite possible that you could use the commands SAM tool like this. So this will help you to convert SAM file to BAM file and BAM file to sorted BAM file. And thus you could avoid using other things. And these things will be done, all these commands could be processed in terminal, the command terminal. Now, it comes to quantification. So generate the number of counts or RPKM values. And this is done by using feature counts or HT-seq software. This will give you all the raw expression values in whole numbers for each and every gene. Once you generate the counts for each gene, then you need to normalize the expression values. You could use any other software, but if you are good in bioinformatics or you have basic mathematics class 10 level knowledge, then you just need to remove most of the values or all the values which are less than equal to 10, and then use log 2 just to convert them into the suppressed values, and this automatically removes most of the outlier values. So the how to use feature counts, it is here the command, and to convert into from this value into log2 values, you just use the command log2 command, and it is easily converted in any language, whether you are using R or MATLAB or any other coding platform. So you have now gene expression values in normalized way or log2 way, log2 values. Then you go for differential expression analysis, where you go for volcano plotting. So volcano plotting is not that big thing, but before you go for volcano plot, you need to go for a statistical analysis first, where you calculate the p-values of each gene, how much significant they are, and then p-value is converted to FDR. Then once you have FDR, then you go for fold change. That means if you are comparing cancer with normal tissues, then you have to predict how many times the gene expression values are higher than normal expression values. That means the genes which are expressed in normal tissues and the genes which are expressed in cancer tissues. So what is how many times they are bigger or lower than each other. So this is how you go for calculating fold change. And this you can do it by using your own basic mathematical formula, and then you choose like plus minus some threshold value, let's say 2.0. This is fold change threshold level. So all the values which are higher than plus 2 will be upregulated. The values which are less than minus 2 will be downregulated genes. So this is what but it is not a kind of a strict threshold level. It could change, go up or down, but there is a rule that it must be higher than one fold change. So, or lower than one fold change. So this is what you need to keep in your mind before you go for predicting differentially expressed genes. So for differentially expressed genes, each gene must satisfy two cutoffs. One is p-value must be less than 0.05 and fold change must be higher than or equal to 0. Plus minus 2. So this is very, very crucial to know. So it is p-values and fold change. So these two things must be satisfied before you could claim that this gene is differentially expressed or not. When you have differentially expressed genes lists, so you need to also prepare the output file for all the differentially expressed genes with expression values in all the samples in cancer and normal tissues, and fold changes in one row and p-values in another row. This will help you to go for figure and analysis, plotting, many more explorations. So it is always wise in each step to have output file in proper way with proper details so you could save your time. Now, the next step is functional annotation. That is how to go for assignment of biological functions. That means the DEX, when you have differentially expressed genes, which means that these genes are altered in terms of expression as a result of cancer. So in terms of function, what are the pathways which are affected? So you need to go for this purpose, you need to go for geoterm analysis, ORCID pathway analysis, or reactome pathway analysis, and there are a number of online servers and softwares which could help you to go for all these analyses. And one of the software which I have mentioned in one of my previous lecture, that is ShinyGo, is very easy to go for all functional analysis. So this is more crucial and important in any kind of high throughput data analysis. And then if you want to proceed for more and more depth analysis, For example, network analysis, then you need to go for network databases such as FunCup, STRING, or PPI network databases, hint. Then there are some more things like IGV. If you want to visualize the genes expressed on different chromosomes, what are the chromosomes where these genes are located, and how to go for plotting, a stylish plotting, attractive plotting. These things are more and more important. So just to analyze is not enough. If you want to publish the research paper or make a presentation in a workshop or international conferences or for your PhD master thesis works, then you need to bring a, the plots or figures of really very good quality and in a smart way presentation that makes you come like really successful in doing the project work. So this is what it meant for visualization tools. So of course it is very a small number of tools which I have mentioned here, but there are 100 times more numbers of tools and softwares for visualization and deep exploration of RNA-seq data analysis. In the next talk, I will be discussing about whole genome sequencing, WES, ChIP-seq analysis, methylation analysis of NGS data. So please be connected and subscribe the channel so it will help you to analyze and perform all kinds of NGS data, and also you could learn free of cost the most of the bioinformatics and system biology and its applications here. Thank you very much.